This lecture is for my Anatomy and Physiology 2 students. This is the last of our lectures, and it will cover fertilization and development. So on to our first slide. Okay, fertilization. What is fertilization? It is the fusion of two haploid gametes and results in the formation of a diploid cell. So the gametes are the egg and the sperm. First of all, the egg. So before fertilization, the egg is not fully mature. It's in a state of suspended animation. It's actually in metaphase 2. Upon ovulation, the egg is surrounded by a layer of cells called the corona radiata. Within this layer is another layer called the zona pellucida. So we have the egg, but we also have to account for these layers that surround the egg. The male gamete is the sperm. The sperm has its own special anatomy, and there is one part called the acrosomal cap. The acrosomal cap contains hyaluronidase and acrosin. It allows the sperm to penetrate the corona radiata and the zona pellucida, those two layers that surround the egg. Now, low sperm count. Sometimes you'll hear about low sperm count. What does that really mean? It's determined that if you have fewer than 15 million sperm cells per milliliter of semen, you have a low sperm count. What does that mean in real life? That means that the chances of you fertilizing an egg may be a little bit lower than others. There are 3 stages of fertilization. Penetration is the first stage. So what does penetration mean? Penetration is when the sperm cell uses acrosomal enzymes to move through the corona radiata and the zona pellucida to get to the egg's plasma membrane. Now, in the previous slide, I mentioned low sperm count, and that low sperm count was fewer than 15 million sperm cells per mil of semen. That seems a high number to be considered a low sperm count. A lot of times you'll see pictures that are similar to what I have at the very, very top. You'll see a picture of an egg, a very large cell, an egg, and then you'll see that egg surrounded by many, many sperm. And here's the thing, sperm cells need their friends. They need their friends. If we can imagine in that acrosomal cap, there being these acrosomal enzymes, and the enzymes, now I want you to look at the picture directly above me. These enzymes have an effect to sort of dissolve the corona radiata and the zona pellucida, those layers that surround the egg. And if you just take one sperm and try to get that one sperm to use its enzymes to move through both of those layers, it's never going to have enough enzyme in order to do that. So that egg being completely surrounded by sperm cells, all releasing their enzymes, actually help out their sperm friends. Okay, now actually one will reach it, right? There's one lucky sperm cell that actually reaches the plasma membrane of that egg. All right, so that moves us into the second stage, and that we call activation. So activation is initiated by the fusion of the sperm and the egg membrane. So the egg has a plasma membrane, it's a type of cell. The sperm is a type of cell that has a plasma membrane. We don't often think of the egg and sperm as being these separate cells, but they are. They have a plasma membrane. And when that one lucky sperm cell reaches the plasma membrane of the egg, their membranes will fuse. Okay, that is what we call activation. Now, upon membrane fusion, the egg's plasma membrane becomes depolarized. You guys know all about depolarization. Okay, so once they fuse, it becomes depolarization. Depolarized. This depolarization initiates the following. It prevents fertilization by more than one sperm. Okay, so once it depolarizes, there's no other sperm that can actually reach it. Okay, depolarizes it, and think of it as it repels the rest of the sperm. Then what happens to the egg? It's going to complete meiosis II. Remember, this egg was in a state of suspended animation. And so at this point, it hurriedly finishes up meiosis II. And then what else is going on in that cell is that you're going to have an increase in the metabolic rate. Okay, I thought this cartoon was cute. I thought you'd like it, kind of apropos for our lecture today. Nope, only one of them is going to make it. Third step in fertilization is the fusion of the nuclei. So the fusion of the egg and the sperm nuclei. We have a technical term for that, and we call it amphimixis. At this point, the cell is now called a zygote. It will have the full complement of chromosomes, which for humans is 46. So if you remember, we think you discussed this in lab about the haploid cells. The haploid gametes, they are 23 because the egg will carry half, the sperm will carry half, and once those nuclei fuse, you have that full complement of chromosomes, which is at 46 for humans. Once fertilization is complete, then we have early embryonic development. So I have a term for you, and it's called cleavage. What does cleavage mean? It means it's a series of cell divisions that continue until the pre-embryo comes into contact with the uterine wall. Now, ideally, the fertilization will occur in the uterine tubes. Then you're going to have early rapid mitotic cell division. Now, when you have that cell division, each cell that results during the series of division, we call these blastomeres. So blastomeres, the cells that are produced by the cleavage. Now, right above me, I do have a picture for you to kind of follow along with. If you want to look at the picture while I talk about these different cell stages. So we can see that cleavage starts to occur, and cleavage really starts on day 1 after fertilization. Day 2, we're getting the 4-cell stage. And then we're talking these cells that are produced by the cleavage are called blastomeres. Now, if we look at about 32 cell stage, it's now called a morula. So the third picture down, that refers to a morula. The cleavages produce a solid mass of cells. So at this point, it's a solid mass of cells. We call it that morula. From there, it really starts to change up, and a fluid starts to form. So once fluid forms, it's called a blastula. And then, looking at the bottom picture there, we have a blastula, but we have this hollow ball of cells. Now, there's different parts to this. We have a trophoblast. So the trophoblast are the cells that surround it, okay? The cells that surround it, and it will provide nutrients to the developing cells. Then we have an inner cell mass, and it's called an embryoblast. So find the trophoblast on the picture and then find the embryoblast. Then we see what's also point to, we have embryoblast cavity. Okay, so there's now distinct regions and differences in that developing embryo. All right, implantation. So we're going to talk a little bit about implantation. Before we get there, I want you to take a look at this picture off to the side, where you see the ovary and the uterine tube, right, and part of the ovary. Now, early on, I said that the fertilization actually occurs in the uterine tube. Optimally, you want fertilization to occur in the uterine tube. Now, what we just learned is once fertilization occurs, then we start seeing cleavage. We start seeing these cells dividing. It happens right away. And it goes through a certain series of development until we get this blastula. Now, it takes some time in order to go from a newly fertilized egg to a blastula. It takes about a week. And as it turns out, a blastula in that form is now ready to implant. Now, could fertilization occur in the uterus between an egg and a sperm? Yes, it could. Could it occur in the part of the uterine tube that is really close to the uterus? It's almost there. Yeah, it could. However, the chance of that newly developing embryo actually being successful with implantation is really low. So for successful implantation, it's ideal to have the blastula. So it takes about a week in order to get to that blastula stage. So anywhere from, let's say, 6 to 9 days to getting to that blastula stage and be ideal for implantation. So if it gets fertilized in the uterine tube, it travels down the uterine tube for 6 to 9 days, and by the time it gets there, it's in a certain stage of development, that blastula, in order to complete its implantation. So now let's talk about implantation. It begins when the blastula comes into contact with the uterine wall. The trophoblast, remember the part that we talked about, that trophoblast secretes hyaluronidase, which allows the blastula to penetrate into the lining. So it physically penetrates into the uterine lining, into the endometrium. And then it creates, it's called a decidua. Okay, so we start calling it a little bit different name. So that particular part is called the decidua. Then we have an amniotic cavity starting to be formed. So we really start to see changes in that developing embryo when it starts to implant into the wall. Now, sometimes we erroneously think that this little embryo is just kind of stuck there and it could fall off. Well, implantation means it's physically, think of it like a halfway burrowing into the wall of the uterus. Okay, that is implantation. Once implantation occurs, then maternal blood starts being directed towards that developing embryo. And once that's established, then some big changes start to occur within that developing embryo. Then we have what's called gastrulation. So gastrulation is the formation of the 3 germ cell layers, the ectoderm, mesoderm, and endoderm. So a little about each of these. The ectoderm, so these are the 3 germ layers. This type, the ectoderm, this is what gives rise to or becomes the epidermis of the skin glands, the nails, sensory receptors in the skin, the cornea, the lens of the eye, the nervous system, tooth enamel. So this is the germ layer that will give rise to all of that. Then we have the mesoderm. So the meso meaning middle, the mesoderm gives rise to the skeletal system, the muscular system, circulatory system, and the lymph. Then we have endoderm. Endoderm gives rise to the epithelial lining of the digestive system, except the mouth and the anus, the epithelial lining of the respiratory system, the liver, and the pancreas. What else we got going on is some extra embryonic membranes. So yes, we have those changes occurring in the embryo, but we also have some extra embryonic membranes, and there are 4 of them. So I want you to know what they are and what their function is. So first of all, the yolk sac. So we have a yolk sac, and take a look and try to find that on the picture next to me. This is something also that can be viewed with the sonogram. So during an ultrasound, you can see the sonogram, and you can see this yolk sac. So you might think, yolk? Wait, we're not chickens. Why do we have a yolk sac? Well, humans do have a yolk sac, but what is it? What is the function in humans? It is important in blood cell formation and development of our blood circulation. Kind of cool. We have another one that's called the amnion. Okay, the amnion is the innermost layer that surrounds the embryo, and it contains the amniotic fluid. So The amniotic fluid allows the fetus to move later in pregnancy, and it provides protection. Now, what type of protection? A cushioning protection for the developing fetus. A third extraembryonic membrane that I would like you to know about is called the allantois. There's actually two different ways that you can pronounce this, allantois or allantois. Both are okay. What does the allantois do? It contributes the blood vessels to the structure that will become the umbilical cord. Okay, so this is kind of like the precursor to the umbilical cord. The fourth extraembryonic membrane is the chorion. Okay, the chorion. What is the chorion? It is the outermost protective layer. It is the part of the chorion that comes into contact with the decidua becomes the placenta. Okay, so we have the placenta that is connected to the umbilical cord, and then the umbilical cord is connected to the baby. Now, when you have the baby, the baby comes out, you have the umbilical cord, and then sometimes what we don't know is that the umbilical cord is attached to the placenta. The placenta also needs to come out. So the placenta will come out, the baby comes out, they clamp the cord, they cut the cord, and then that placenta comes out as well. So that whole entire structure comes out. Where did all those things come from? It came from these extra embryonic membranes, these membranes that surrounded the embryo even from the get-go. All right, guys, that's it. This is the last of our lecture. I hope you enjoyed it.